Chapter 11. Your Happy Genes. Based on what we know of the plasticity of the brain, we can think of things like happiness and compassion as skills that are no different from learning to play a musical instrument or tennis. It is possible to train our brains to be happy. Neuroscientist Richard Davidson. Wouldn't it be nice if we had a set of genes that coded for the biochemicals of happiness? Imagine if every one of us possessed a gene collection that, when the right genes were turned off or on, made us happy. Being happy is what virtually everyone says that they want. Even miserable people seem to be happy being miserable. Many human activities revolve around the goal of creating the conditions in which happiness can flourish. Material well-being, safety and security, and physical health all contribute to happiness. Spiritual practices can change our brains in ways that produce happiness. Yet happiness is not an on and off condition like a light switch. You aren't either miserably depressed or ecstatic, with nothing in between. It's more like the dimmer on a lamp, in which the position of the slider might be at any point between total lightness and total darkness, and is usually somewhere between those two extremes. In this way, it's like our fight or flight response. We aren't either in total fight or flight mode or totally passive. Most of the time, we're somewhere in between. Another spectrum is the production of cortisol and DHEA in the cortex of our adrenal glands. Your body isn't going flat out producing either one or the other. It's producing a ratio of both hormones. Prolonged stress will lead you to produce much more cortisol. Prolonged relaxation will lead you to produce more DHEA. At any given moment, you're somewhere between the extremes. Happiness can also be found along a continuum. At any given moment, we're somewhere on the spectrum between ecstasy and despair. The difference between feeling slightly content and slightly discontent might only be a few clicks along the slider, not very far apart at all. The trick that happy people have learned is to push their emotional state closer to the happy end of the scale and further away from the dark side. How do our genes figure into these endlessly variable emotional states? And what can we do to nudge our experience closer to the light side of the spectrum? Genes and emotional states. When emotional states occur in the psychospiritual realm, biochemical changes occur in the body. These biochemicals are the products of gene expression. If your body is building more white blood cells like AIDS patients who have spiritual transformations, or if you're boosting your immune markers like people who master the art of producing coherent heart rate patterns, the genes that code for these physiological changes must be active. When we measure the boost in a feel-good neurotransmitter like serotonin that occurs during positive experiences, it stands to reason that the underlying genes that tell our cells how to produce serotonin must be expressed. Researchers have now published a sizable number of reviews of studies that show a link between emotional states and genes. They've examined the correlations between emotional disturbances like schizophrenia, depression, panic, and post-traumatic stress disorder, and gene expression, especially in the brain. They have come up with startling and compelling evidence that the patterns of gene expression in the brains of these unhappy sufferers are different from those of happier people. Emotions, it turns out, are epigenetic. and can methylate genes in parts of our brain involved in the stress response. The process is not a one-way street, with emotions turning genes on and off. People with certain genes are more prone to unhappy emotions. Though the evidence suggests that most conditions require external epigenetic triggers to stimulate these genes, a 2008 study funded by the National Institute of Mental Health found that there are genes that predispose a person to post-traumatic stress disorder as an adult. But PTSD is much more likely to occur in these adults if they also have a history of abuse as children. It's an external environmental event in the form of a traumatic childhood that provides the epigenetic stimulus to the inner environment that triggers the gene changes. Such studies, using the new technology of DNA microarrays or gene chips, are allowing scientists to peer into the gene expression in the fight-or-flight structures of the brain and find out how genes are different in people with various flavors of unhappiness. Unhappy emotions, unhappy brain. Moshe Saif, Ph.D., discovered that nurturing by mother rats provides an epigenetic stimulus to their offspring. Nurturing alters the expression of genes in the hippocampus and other parts of the brain that govern the fight-or-flight response, giving nurtured rats a better ability to manage stressful stimuli. This made Saif curious about whether human brains would show similar changes. He speculated that schizophrenics who had suffered abuse during their childhoods would also show changes in the hippocampus too. He dissected the brains of 24 people who had donated their organs to science. 11 of these had happy childhoods. The other 13 were very unhappy indeed. They were schizophrenics who had committed suicide. Saif found that many of the stress-dampening genes in the hippocampi of the unhappy people were switched off, and although the unhappy people possessed all the right genes to dampen their stress response, the genes were methylated, which suppressed their activation. A Canadian study using brain tissue collected during autopsies also found that gene suppression in the brains of people who had committed suicide was much more prevalent than in happy people who had died accidental deaths. Saif calls these suppressed genes frozen assets because while they are present in the brain, they're not accessible to its owner. In a part of the brain not involved with mood, called the cerebellum, there were no significant epigenetic differences between the happy and the unhappy groups. This suggested that the methylation of the mood area was a response to the unhappy childhoods that the schizophrenics had suffered. In a study of 177 boys incarcerated in a juvenile detention center, psychological depression plus a gene that transports the stress neurotransmitter dopamine were linked to unhappy and neglectful mothering. Another researcher compared pregnant women who were being treated for depression using SSRI drugs, a class of antidepressants, with women who were depressed but not receiving the drugs, and also compared them to happy women. They found increased secretions of stress hormones like cortisol in the babies 3 months after they were born, with a corresponding suppression of the infant's stress-dampening genes. This research provides a potent reminder that it's not the presence or absence of genes alone that determine whether they build the proteins for which they designed. We can suppress or facilitate their expression by external environmental influence. Stress-induced genetic changes. What's particularly interesting is to compare the genes of the same people before and after a stress experience. One study compared the gene expression of medical students to their anxiety scores on a standard psychological test. A measurement of their gene expression was taken just before their licensing examinations, a time of high test anxiety for the students. It was compared with a second gene chip sample and psychological test taken 9 months earlier to provide a baseline. The researchers found that during the high-stress period, 24 genes were expressed differently. The expression of stress genes tracked the students' scores on the psychological test. Another set of researchers looked at people who were lonely and depressed and compared the expression of their entire genome with a happier group. They found 209 genes that were differently expressed in the unhappy people. The nature of the affected genes is particularly interesting. They code for, amongst other things, markers of our levels of immunity from disease. So unhappy people had weakened immune systems and were much more susceptible to illness. A research team found that depression and elevated cortisol correlate with changes in the expression of many of the genes in the limbic system of the brain, which is central to our stress response. Another group examined the link between a gene that codes for cortisol and other aspects of the fight or flight response and the psychological trait of anxiety. Anxious children were more likely to have this gene strongly expressed. The children of parents who had panic disorder, a very high degree of anxiety, showed an even stronger association between behavior and gene expression. To translate what the researchers are saying into plain English, a language carefully avoided in scientific papers, the crazier and unhappier your family is, the more likely it is that you'll be anxious and unhappy yourself, with the gene modifications to prove it. Not only do your genes change with unhappiness, your brain wiring changes too. Jeffrey Schwartz, Ph.D., a neuroscientist at the University of California at Los Angeles, has studied a psychological illness called obsessive-compulsive disorder, OCD. He's tracked the changes that occur in bundles of neurons in the brain when OCD is treated. Successful treatment results literally in a rewiring of the neural connections inside the brain, like a house that adds wiring to the electrical connections that are used most frequently and strips wiring away from neglected circuits. The plastic brain is in constant motion, responding to stimuli by creating new neural pathways. Unhappiness reinforces our unhappy brain wiring and vice versa. Science is catching up to where spirituality has been for thousands of years. The Buddha urged us to maintain a calm state of desireless attention to the present moment, reminding us, we are what we think. All that we are arises from our thoughts. With our thoughts, we make the world. Psychologists sees different diseases. Biologists sees similar disease. It's interesting to notice that these epigenetic signals were provided by a wide variety of unhappy psychological conditions. Test anxiety is different from loneliness, which is different from PTSD, which is different from schizophrenia, which is different from OCD. Yet the body seems to interpret them all as similar epigenetic stress signals. Psychology has made an art of identifying and differentiating psychological conditions. The Diagnostic and Statistical Manual of the American Psychiatric Association, currently in its 4th edition, the DSM-IV, notes hundreds of classifications of distinct emotional maladies, from vascular dementia with delirium, 290.41, to factitious disorder with predominantly psychological signs and symptoms, 300.16. Although reductionism, the act of differentiating mental disorders into so many categories, has its uses. It's worth remembering that the body doesn't work that way. Whereas all these flavors of stressed out unhappiness may look different to a psychologist analyzing the mind, they look biochemically similar to a biologist analyzing the body. Now, the term stress was coined by German physician Hans Sly in the 1930s. As he walked the wards of his hospital, he noted that most sick people had certain common symptoms, including aches and pains, looking and feeling unwell, digestive upsets, rashes, and fever. He called this syndrome stress. When there is enough environmental pressure, whether it's physical, emotional, electromagnetic, or chemical, to produce a surge of adrenaline and cortisol in the bloodstream, then what Sly called an alarm reaction follows. Sly went in exactly the opposite direction from the reductionist approach in the DSM-IV. He didn't seek the highest common multiple of symptoms. Instead, he identified the lowest common denominators. Now, this makes evolutionary sense. When a woolly mammoth charged at your distant ancestor, your ancestor fought back or ran away. Anyone who hesitated was maimed or killed. Any one of 1,000 different environmental epigenetic inputs, from a snake to a thorn to a scream, to a lion to a spear to a poison ivy bush, would result in a single epigenetic output: activate, fight, or flight. When the woolly mammoth appeared, your ancestor didn't sit down on a log and wonder, gee, is this a specific phobia 300.29, a panic disorder without agoraphobia 300.01, or mood disorder 296.90, and then decide which course of action to take based on that diagnosis. He simply got out of Dodge as fast as his legs could carry him. Stress drives gene expression. Once Hans Sly had developed his insights into stress as the common denominator to much illness, a Harvard physician became famous for asking the logical next question: how can we reverse stress? His name was Herbert Benson, and he developed a method called Relaxation Response. This stress antidote had practitioners sit quietly for 20 minutes, filling their minds with a positive phrase or belief, and focusing on relaxing their muscles from the feet all the way up to the head. Studies showed that the relaxation response was able to help with many different ailments, from high blood pressure to infertility to rheumatoid arthritis to pain. How could a single, simple stress reduction technique affect so many different conditions? Researchers wondered. The answer had to wait until the invention of DNA microarrays at the end of the 20th century, which made possible the study, the relaxation response, as an epigenetic intervention. Benson took healthy subjects and compared the differences in gene expression patterns between long-term practitioners of the relaxation response and non-practitioners. Then he put the non-practitioners through an 8-week training and found that their gene expression profile had changed to substantially resemble that of the long-term practitioners. Then he replicated his findings before publishing them. Among the genes that changed were those involved with inflammation, the rate at which cells regenerate, and the scavenging of free radicals, which are a prime contributor to aging. For hundreds of years, Western medicine has looked at mind and body as totally separate entities, to the point where saying something is all in your head implied that it was imaginary, said Benson of the study. Now we've found how changing the activity of the mind can alter the way basic genetic instructions are implemented. The Benson Studies co-author says, "This is the first comprehensive study of how the mind can affect gene expression, linking what has been looked on as a soft science with the hard science of genomics." Psychology and spirituality have been regarded as less rigorous than biology and physiology, and the scientific world is having to rethink its priorities as it discovers that invisible human emotions have profound epigenetic effects. Dean Ornish, Ph.D., who made the cover of Time magazine for his groundbreaking discovery that diet can reverse coronary artery disease, has taken his insights one step further in a gene study published in 2008. He examined the effects of lifestyle change, including an hour a day of stress reduction technique, such as meditation, on men with prostate cancer. The subjects had decided not to undergo conventional surgery and radiation or hormone-based cancer treatments. Instead, for 3 months, they changed their lifestyles. In addition to meditation and other stress management methods, they walked for half an hour a day, ate a diet rich in legumes, soy, fruits, whole grains, and vegetables. It came as no surprise to the researchers to learn that the men lost weight, lowered their blood pressure. But when the researchers compared prostate biopsies taken before and after, they discovered that an astonishing 501 genes had been changed in just 3 months. Genes associated with breast cancer and prostate cancer had shut down. Ornish observes, "It's an exciting finding because so often people say, 'Oh, it's all in my genes, what can I do?' Well, it turns out you may be able to do a lot. Meditation, prayer, spiritual practice, and attendance at a house of worship are all stress-reducing activities. A vibrant spiritual, social life also affects lifespan. The hospitalization rate of churchgoers is half that of non-churchgoers." Stress, emotions, spirituality, and disease are intrinsically linked. Scientific papers have now documented the connection between stress and many organic diseases, including heart attacks, cancer, and the progression of HIV and AIDS. Emotional states such as panic, hostility, depression, and anger all contribute to heart attack risk. The link between cancer and stress is particularly interesting and has been shown in much research, but a recent study found receptor sites on the outside of cancer cells for adrenaline, indicating a straight-line biochemical link between stress and tumors. And when we're stressed, our bodies are flooded with cortisol and adrenaline, and this study found that cancers grew 275% faster in stressed mice than in unstressed mice. The scientific consensus is that only 5% to 10% of cancers are hereditary. The rest are due to environmental factors including stress. In a paper entitled Spirituality and Immunity, Stephen Arini, Ph.D., lists studies that show the following results of stress at the cellular level. Wound healing takes 9 days longer. Intracellular communication is decreased. The immune system is suppressed. Poorer inflammation response at the site of injury. Increased bacterial infections. Increased susceptibility to autoimmune diseases. Decreased ability to handle invading toxins. We don't have a pill for this, says Moshe Frankel, MD, Medical Director of the Place of Wellness, the Integrative Medicine Department at the MD Anderson Cancer Clinic in Houston, Texas, one of the most advanced cancer treatment facilities in the country. But we do have yoga, meditation, and guided imagery. Using a de-stressing technique doesn't just activate your happy genes, it activates your healthy genes too. Telomeres and lifespan. Many of the genes found in these studies were regulatory genes, regulating the expression of other genes and proteins. Regulatory biochemicals can affect thousands of other genes and the synthesis of regulatory proteins and neurotransmitters. In fruit flies, PAX6, a regulatory gene that plays a role in the development of the insect's eyes, has been artificially activated under laboratory conditions. When the expression of PAX6 is induced on the antenna of a fruit fly, it triggers the expression of some 2,500 other genes, leading to the development of an extra eye on the antenna. So the regulatory genes are powerful, and the effects on the body go way beyond the changes in a roughly 1% of the genome noted in the group of studies with human subjects summarized previously. Telomeres are the tails of genes, and they get shorter each year from birth to death at a constant rate that averages 1% a year. They are a marker for aging. The older the person, the shorter the telomeres. Bad lifestyle habits accelerate the process, and good ones slow it down slightly. But a pilot study by my friend and mentor Norman Shealy, MD, PhD, has shown that the process can be reversed in human beings. Norm is the founder of the American Holistic Medical Association and the co-author with me of the book Soul Medicine, which explains the world of energy medicine and how to find and work with your ideal practitioner. In his study of the epigenetics of aging, Norm measured the telomeres length of 6 subjects, then put them on a healthy diet and exercise routine, including his patented Rejuva Matrix, which produces an electromagnetic field with the same frequencies as human DNA. After 3 months on Norm's program, the telomeres of his subjects had stopped getting shorter. They actually began to lengthen. After 10 months, they were 2.9% longer, suggesting that we can reverse many of the signs of cellular aging by a healthy electromagnetic environment and a supportive lifestyle. Analysis of 30 studies by Dutch professor Ruud Vinhoven found that happiness adds between 7 and 10 years to your lifespan. He found that happy people were more likely to control their weight, perceive illness symptoms early on, and moderate their smoking and alcohol consumption. Chronic unhappiness activates the fight-or-flight response, which is known to involve harmful effects in the long run, such as higher blood pressure and lower immune response, Vinhoven observed. Not only is happiness healthy, it's contagious. You can catch it from the people sitting next to you. A large-scale study of residents of Framingham, Massachusetts from 1983 on noted their degree of happiness, amongst other things. Since they had such a large group, the researchers were able to analyze how happiness spread throughout the population. They found that having a friend who's happy increases your chances of happiness by 15%. A second-degree connection, such as the happy spouse of a friend, raises your chances by 10%. And a third-degree contact, such as a friend of a friend, increases it by 6%, after which the effect trails off. And surrounding yourself by more happy friends increases the effect. Since our social networks are largely under our control, this research presents us with yet another practical leverage point over our gene expression. While dumping negative friends and seeking positive ones may be difficult after a lifetime of association, it's a strong signal of an intervention to recreate our lives in a healthier mold. And if you find positive people avoiding you After they read the Framingham study, you might be one of the unhappy people they're avoiding, and may need to undertake what Alcoholics Anonymous call a fearless moral inventory. Choices reverse childhood. If you had a lousy childhood and your stress-dampening mechanisms were epigenetically compromised, you're doomed to a lifetime of unhappiness? Here again, research has some encouraging answers for us. When scientists took mice that had been nurtured early on and deprived them as adults, they undid most of the positive effects of maternal nurturing. But when they took the mice that had been neglected as newborns and provided them with a nurturing environment, the deficits they had shown were partially reversed. Other studies reflect the same possibility. One found 27 genes upregulated by a hearty dose of laughter. This suggests that it's never too late to give yourself a happy childhood. Surround yourself by nurturing people, and conditions can start to reverse the methylation and acetylation patterns in your stress system. Every minute, about 1 million of your cells die. An equal number of cells are born each minute. Perhaps you implanted lousy epigenetic signals into your old cells through destructive habits and emotions. But your old cells are on their way out. If you start sending new epigenetic signals to the cells that are just getting born, filling your heart with love and your mind with positive thoughts, your new cells might be healthier than the ones they're replacing. Every single molecule in your brain is replaced every 2 months. Every single cell in your body is replaced every 7 years. So as you give yourself a psycho-spiritual makeover, you give yourself an epigenetic makeover too. This kind of genetic drift over time shows up in identical twins. They start out with exactly the same genome, and at the age of 3, their genes are indistinguishable from one another. But by age 50, there are many differences in methylation, and hence gene expression. They have made different emotional and spiritual choices. Their lifestyles are the habits that they've cultivated. They have different stress levels, and every cell in their body has been replaced at least 7 times. Though they began life with the same genome, an average identical twin dies more than 10 years apart. And that's a huge difference, indicating that the choices you make have powerful epigenetic effects at all stages of your life. So the scientific evidence continues to pour in, and it is compelling. It tells us where we are on the happiness scale. Our emotional state has a powerful epigenetic effect on our bodies, affecting all the major systems of our body. It also provides us with a list of the practical things we can do to nudge ourselves toward happiness. We can use positive beliefs, spirituality, prayers, positive images and visualizations, electromagnetic shifts, optimism, intention, energy medicine, energy psychology, positive attitude, nurturing, gratitude, happy social networks, acts of kindness, meditation. The exciting implication of this research is that so many of the items in this list are under our conscious control. We can change our attitude by an act of choice. We can stop ourselves speaking an unkind word. We can volunteer for a charity. Every day presents us with opportunities for altruistic acts, if we're awake to the possibility. We can say uplifting words to the people around us and perform small acts of service. We can pray to whatever deity speaks to our deepest longings. We can meditate. We can join a house of worship, a club, a sports team, or a support group. We can use energy medicine and energy psychology to quickly dissipate the emotional charge of traumatic events. We can invent stories and imagine visualizations that are positive. We can turn off our TVs and read books that enrich our hearts. We can watch inspiring movies and listen to soothing music. Although these conscious-based methods are different, they all can affect the stress response. Vinson says, "We found that no matter which particular technique is used, different forms of meditation and yoga, breath focus, or repetitive prayer, the mechanism involved is the same. As we use these conscious-based techniques to shift our emotional state from anxiety to serenity, we affect our gene expression. These are all behaviors that we can choose deliberately, inducing happiness and giving us active control over the genetic markers that we place on the game board of life. Your happy genes are present in your body and brain as potentials, and what you do with them shapes your destiny."